So yes, the recorder is on this morning. At least it certainly looks like it. I was so careful on Friday to make sure I had batteries for the mic and then forgot to turn on the recording. Sorry about that. But I did post last year's lecture, which is very similar in terms of the content. There's some really great stuff about MERS, the Middle East Respiratory Syndrome, right at the end of the lecture that we didn't have a chance to go over, so it's not going to be stuff which is on the exam, but really important to know about, I think, anyway. But again, these are not things that will be on the exam. So today, again, just finish up really quickly, review positive strand RNA viruses. These are the classic coronaviruses. Again, the largest, in fact, of any of the RNA genome-containing viruses. Only originally known as the common cold, and then, of course, SARS, and now more recently MERS came along, and we had all of this, excitement, I guess, is one term for it, in terms of thinking about these coronaviruses and particularly coronavirus diseases. So do we have any questions from the audience, as it were, while you get out your clickers? So now's your chance. Yes, happy, no. So this is what happens if I have extra clicker questions at the end that I don't get to. I can put them in the next lecture, which is great review, I think. You may disagree with me. Oh, by the way, I am going to try and do a sort of midterm evaluation since it's about halfway through, so you can tell me what you like or don't like about clicker questions, random roll call, etc. And I will at least take a look at your answers. Okay, all settled in the back? All got our clickers out? Okay, we'll do our normal thing here and let everyone take a quick guess and then have a chance to, if we get it to start, yes. And then we can discuss what you think your answer should be relative to your neighbor. And again, we can aim for 100%. Hopefully, at least one of the clicker questions today is going to be 100%. I'll let you guess which one it should be later on. And again, if we get 90% the first time around, we don't need to go to a second round. But I know you all want to talk about it, so. Nope, not 90%. Okay. Tell each other what you decided and why. Okay, it's getting quiet again. Caffeine's wearing off. Let's try again. If any of you have been looking at old exams, you will notice this is one of those kinds of questions that I love to ask. Vote early, vote often, right? So, as usual, it looks like someone's trying to ruin my statistics. I'm not quite sure why, but that is your own choice to be able to do that. So yes, the coronavirus structural proteins are at the 3' end of the genome. Why is that different relative to anything else that we've talked about so far? Because all the other positive strand ones have always been at the 5' end of the genome. So structural proteins for flaviviruses, for picoRNA viruses, etc. And then for MS2 and Q beta, kind of spread throughout because there's the maturation protein all the way at the 5' end and the coat protein kind of in the middle. So this is great because, again, I can be lazy and use the same answers for lots of different questions. Oh, hang on, let's choose the right answer. That would be good. Oh, and that does remind me, I haven't uploaded the clicker points for a while yet. Some of you may have noticed that. I will try and do that at the very latest later this week. Okay, so today we will go over to the other side, negative strand RNA viruses. So again, and this is what we'll do, negative strand RNA viruses up through the next midterm, coming up all together too soon. I got to write another exam. I know you wouldn't be fine if I didn't write it, I guess. But so these negative strand RNA viruses, we'll talk about the MonoNegaviruses, which are particularly today, and what we'll talk about on Wednesday. And then we'll talk a little bit about flu, some of these segmented ones. As I mentioned way back at the beginning of the course, these are some of the nastier diseases that we have had to deal with. So measles, mumps, respiratory syncytial virus, rabies, and then more lab rats, as it were, pun not intended, particularly with Sendai because it's a mouse virus, and vesicular stomatitis virus. These are ones which don't make humans sick, but they're incredibly useful tools for understanding how these viruses work, and in the case of VSV, also a really useful vaccine platform. A couple of key concepts with these MonoNegaviruses are these so-called subgenomic RNAs. Subgenomic RNAs are those, again, we talk about structural proteins for the coronaviruses. How are they made is different. Again, in the coronaviruses, these nested subgenomic RNAs. For these MonoNegaviruses, they have this, what they call start-stop messenger RNA production, which is really kind of funky as far as any other kinds of messenger RNA production that probably any of us have thought about before this. A lot of that has to do with this process called stuttering, which is literally the RNA-dependent RNA polymerase making multiple copies of one or a few nucleotides as it's transcribing. We haven't talked too much about alternative start codons, but it turns out for many of these viruses, it's another way to use the genetic material that you have in lots of different reading frames. So it's a genetic economy. Again, mostly for these viruses, smaller is better. There's a small amount of RNA editing, which takes place in some of these viruses, probably not as critical as we'll see in later viruses. And then the other one, which I just added here, vaccinate, 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 repeat ad nauseum. So as usual, little outline, origin. Again, this is where we'll talk a little bit about disease and the fact that there's not so much disease from a lot of these viruses, again, because of vaccines, where they came from, and then the structures of some of these viruses. They're actually kind of blobby, at least most of the Paramyxoviruses. The Rhabdoviruses, on the other hand, have this interesting called bullet-like structure. How do they get in? Turns out this is a pretty standard mechanism, and turns out to be extremely useful, again, for some of these vaccine purposes. Replication, this is probably where I should have put the red because this is where we have the differences relative to some of the other viruses so far. Translation is pretty straightforward. release is pretty straightforward. So what about these MonoNegaviruses? Again, the virologists have ridiculously creative names for these, and at least they don't try and call them MonoNegaviruses or something like that. So just single genome, negative strand. And again, for this lecture and Wednesday's lecture, we'll be talking about these particular viruses. So again, not surprisingly, it's a single negative strand. And as we'll see for flu, that's multiple negative strands. And there are quite a few other ones. Some of the other chapters in the textbook talking about some of the Bunyaviruses, the hantaviruses. These all have multiple genome segments. So these are different, and they just have this single genome strand. They all bud at the plasma membrane. And of course, negative strand viruses need something in particular, which is they have to have their own RNA-dependent RNA polymerase in the virion. So since negative strand by definition means that it's not coding sequence, then we have to have that protein in the virion when it comes inside the cell. And then again, this just start-stop messenger RNA production. So how do we learn about these kinds of viruses, particularly the rhabdoviruses? Rabies, one of the, I guess, most famous, one of the nastier viruses around in terms of disease. If you do contract rabies from usually a rabid animal, you will die. I think there have been like 1 or 2 exceptions to that. In terms of all the people who've been infected with rabies. So it's very, very nasty. It's encephalitis. Your brain just swells up. Not good. I just heard actually I think it was a tweet that I read. Measles is for a week, encephalitis is forever. So yeah, not a good thing. But one of the great things about rabies, as far as treating the human disease, is that the progression is relatively slow. So you can actually vaccinate after you've been exposed to the virus. And it's one of the very, very few examples of this. And one of the very first vaccines to be produced. And I added this thing down the bottom here because a few people have asked me about how you get attenuated vaccines. And Pasteur is really credited with the first set of these attenuated vaccines. And so what he, which means probably the people he told to do this, did was inject rabbits with a really nasty rabies virus, collect, in this case, the spinal fluid from these rabbits, inject it into another rabbit, take that spinal fluid, inject it into another rabbit, and do this many, many, many times. And hopefully you'll notice the syringe is getting greener and greener. And that is that this virus now, at least as far as humans are concerned, is less and less pathogenic. And so the original rabies virus was, in fact, made in this particular process, and in fact given to a poor boy who had been bitten by a rabid animal. And you may say, okay, what about clinical testing and so on and so forth? Well, if it's a completely fatal disease, then people would also call this more of compassionate use. And again, this is far before vaccines, viruses were known as viruses. In fact, it's probably the second vaccine after Jenner's vaccine that we'll talk about when we talk about smallpox later on. So this is rabies, again, the classic rhabdovirus. Not surprisingly, most people don't want to work with rabies in the lab, and so there's a nice analog of this, the fascicular stomatitis virus. Mostly infects livestock, but it's become kind of a standard model for studying these kinds of rhabdoviruses for a couple of different reasons. One of them is it has an extremely broad host range, which turns out to be very, very useful for development in vaccines. And we'll see why that is a little bit later on. And this is great for vaccines, but also people are using vesicular stomatitis for gene therapy. Why? Because what do viruses do? They take their genes in the bag and put them inside a cell. Well, if you're trying to do gene therapy, what do you want to do? You want to take a gene and put it inside a particular cell. And so that's the idea here, and in fact a lot of gene therapy processes are in fact using VSV, and we'll talk about some other gene therapy later on. But probably the most famous application, at least recently, of VSV is this VSV-ZEBOV. What the heck is ZEBOV? Zaire Ebola virus. So this is the current mostly used Ebola vaccine. This was put into clinical trials in West Africa in 2014, which was very soon after the outbreak, which we'll spend a long time talking about on Wednesday. And this is pretty typical in terms of the kinds of clinical trials that are done. First, a phase 1, which is a safety trial, a phase 2, which is about optimizing protection and safety, and then phase 3 to actually see if it works. And this turns out to be a pretty major issue as far as Ebola vaccines are concerned, because there's not much circulating Ebola. So it's, in fact, a really good thing that there's not too much out there, but it means making these efficacy trials very difficult indeed. So this is a very standard process, and we'll talk about this again when we talk about Ebola virus disease later on. But phase 1 is usually under 100 people. Phase 2 is hundreds to thousands, and phase 3 is thousands to tens of thousands. And again, this is really typical for any kind of drug trial. The last one of these was done in March 2016, a so-called ring vaccination study. To do these classic kind of clinical trials, you want to have placebo and your effective drug, but if you've got something like Ebola that kills 20 to 30% of the people it infects, you really don't want to be using too much in the way of placebo, particularly if there's an outbreak. And so there are all kinds of different ways that you do these vaccination trials. So this particular one was a ring vaccination, basically earlier and later treatment of people who would have been exposed to the disease. All of them ended up getting this particular vaccine. And it was reported 2 years ago, almost 2 years ago now, that 70 to 100% efficacy of this vaccine, which is really pretty amazing that the vaccine worked so well. This is a recombinant vaccine. That's what this R at the beginning here stands for. And again, you can think about what the recombinant nature is of these vaccines a little bit later on. So again, VSV, very wonderful platform in terms of using them for vaccine purposes. The Paramyxoviruses, which I'm going to spend the next 15 minutes or so talking about, main one here is measles. There are also mumps, Nipah and Hendra viruses, and Sendai viruses. Nipah and Hendra are new Paramyxoviruses emerging mostly in Southwest Asia, probably mostly because we've been cutting down too many of the forests, and a lot of the animals that normally were reservoirs for these diseases are spreading them to humans. So pretty classic kinds of issues there. Measles has been around a lot longer, and we'll talk about the process towards hopefully eventually getting rid of measles. Rinderpest is the sort of measles equivalent for Rind. Anyone speak German? Rind is cows. So it's a cow disease which has actually been completely eliminated due to vaccination. So sort of smallpox was one of the huge successes in terms of human vaccination, but Rinderpest is great success in terms of the agricultural processes. So why has this worked so well? Because there are great vaccines for these. And this is me getting on my high horse here, and I'll trumpet this again for a lot of the rest of the course. Get your measles vaccine, or if you're a parent, make sure your kids get it. It's part of the measles, mumps, and rubella vaccine. There are lots of good reasons to have them together, which we're not going to talk about here, but I'm happy to talk about offline. And why? Because 0.1 to 0.2% of kids who get measles die. That's a really scary number. But as we'll see, unfortunately, or fortunately, depends on your point of view, we're right at that kind of borderline now, at least in the US. Between 500 and 1,000 kids every year end up getting measles. And everyone's, oh, kids don't die from measles. Yeah, kids do die from measles. And in fact, in Europe, a few kids have died from measles in the last couple of years. 1 in 20 of these kids who get the measles will get pneumonia. This is probably what causes most death. and also in the same kind of percentage-wise, get encephalitis, which can lead to deafness, intellectual disability, et cetera. And if you know Roald Dahl, the children's author, his daughter died of measles and encephalitis before there was a vaccine. Now, there is, and this is something I think which is really important, again, talking to the pro-diseasers, there is a certain amount of risk involved with vaccines. No question about it. I'm not going to say they are absolutely 100% safe. It's not 100% safe for me to be standing here in this building in an earthquake zone. So there, 4 in a million will get serious allergic side effects. Now, 4 in a million versus 1 in 1,000. So you can do the math. I did the math for you. So again, in theory, it's your choice, although people have now started some legislation to say that actually the kids should have the rights as opposed to the parents deciding for them, and that's another interesting issue that we can, again, talk about offline. But a lot of this is kind of an out of sight, out of mind issue, I think, with a lot of the, again, I'll call them pro-disease people. Before vaccines, and we'll see some statistics about this in just a second, there were 4 million cases in the US. And again, you can do the math, 4 million, 1 in 1,000 dying, lots of kids were dying. And measles, because of this great vaccine, was quote unquote eliminated in the US in 2000, but as some of you may have noticed, there have been 35 in Missouri and Kansas since March of this year. So it's not completely eliminated in the US, and as we'll see a little bit later on, almost all of these cases have been imported because measles is not eliminated in the world, and we like to travel to various different places, some of which we'll see in just a second here. So here's some of the data on this pre-vaccine, post-vaccine. These are thousands of cases per year, again, up to almost 1 million per year in the late 1950s. In the 60s, the vaccine was licensed and goes down to tens of thousands in the late 1960s. Then we started with a second dose. Now we've changed our scale completely here, 5,000, and then there was this mini outbreak getting up to 25,000. Again, 25,000 still here is just a tiny blip. This is this tiny blip right here. And with this vaccination gone to nearly zero, at least on this kind of scale. But unfortunately, it's stuff which is coming back up. Again, not unlike the case now with vaccine-derived polio. It's not the US and not spreading here because most people, fortunately, are vaccinating. But there are certainly introductions from other places in the world. So this is the clicker question coming up that everybody should, before Jacob. This is not vaccine-derived measles, no. It turns out that for reasons that are not entirely clear, the measles vaccine, which is made as an attenuated vaccine, doesn't seem to revert, which is actually kind of bizarre that polio seems to revert and measles doesn't. Quite why that is not entirely clear. May have to do with the way the attenuation process happened, that those mutations, if they reverted, then you would actually lose some kind of activity. They may be interacting with each other. It's a great question because people would really like to know sort of what that mechanism is. These were, again, developed in the early 1960s when we didn't have as good handle on exactly what those changes were and what was going on with them. So people are developing sort of some next generation measles vaccines that should have absolutely no chance of reversion, but interestingly enough, these don't seem to revert. Yeah?
On the last slide, you mentioned that there were vaccines that were about 4 million
Yeah.
per year. But on the graph, it's not.
Yeah, and so this is 1,000. These are all different kinds of ways of doing these different calculations. And this number is actually from the book. And these are numbers which I pulled up from some of this public domain commons on Wikimedia. This is probably a better number than that previous one. And so if Stedman were to ask a question about it on an exam, he would have to be very careful about which number he used. Because he loves numbers. Why does he love numbers? Because they're easy to write answers to. Coming up with the wrong answers. That's why he loves numbers, because he's lazy. OK, so we're all happy. Speaking of numbers, yes? OK. So this one, hopefully, we'll get 100% on the first time, right? Except for the people who are trying to ruin my statistics. But again, if we get more than 90% the first time, I'm going to stop it and we won't re-vote again. So. Last chance to vote. 3, 2, 1. Pardon? So we've got, let's see, you can do my calculations correctly. What are we at? 89%, I guess? 87%. But I'm going to stick with that. So whoever voted for A, we should talk after class. And the rest of you, we can discuss about this later. So the answer is yes, E. OK, more numbers. Again, Stedman's loves numbers. But this is mostly to say how important vaccines are and how useful they are. And that is, there were, and this also is different than the numbers in the textbook, according to the WHO, about 500,000 deaths, again, worldwide now in 2000. When we had 72% of children vaccinated, that's been going down over the years. And 2016, which is the last year, we have numbers for, 85% of kids have been vaccinated, and we're below 90,000. That being said, there are still many who are infected in any given year, and you can look at the numbers here. And the WHO says that hopefully by 2020 it will be eliminated in 5 different regions of the world. And these are the WHO regions, which I forget exactly what they are, but they're kind of like continents, if I remember correctly. This, I think, is unfortunately a little, given that we're in 2018, seems a little unlikely. So hopefully after polio, this will be the next one of those diseases that we'll actually be able to completely eliminate. So, can I get this to move? Come on. OK, so this is, again, I tried to find a more up-to-date map on this. This is actually last year's, getting up to February, but it's pretty similar this year as well. Some slight changes. Hang on, we're off here. OK, my phone is again causing me problems here. OK, we'll switch back over. Here, the number of reported measles cases, again, in about 6 months. Most of them, and now actually mostly in India, which is probably the place where there are the most measles-related diseases now. But certainly other places in the world, and particularly those like North America, where we're still getting, in this case, it was hundreds, or actually under 100. This year, actually, that should be a little bit more red, because we've had more than that. And it's really unfortunate that there are places where there's good vaccines that we're still seeing many cases. Romania here, you notice this dark red blotch here. That's where there have been a number of kids who have died of measles, literally in this last year. Because we've got more than 1,000 cases, 1 in 1,000 of them are unfortunately going to going to die. So vaccinate, 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 and this should be really obvious, but unfortunately, we have other things that are happening in various countries, including ours. 2008, practically fairy tales before the, sorry, I crossed this out, pro-disease movement. Practically no measles, and in 2014, large measles and whooping cough outbreaks. These ones we're going to look at in a little bit more detail here, particularly 2014, 2015 being some of these major outbreaks. But again, I am kind of hardcore about this as far. Oh, yeah, Jacob. So in terms of this here, this is probably actually underreporting in some cases, but also there seems to be better vaccination in many of these places than there are in other places, potentially due to this issue. Some of you may have remember from Dr. Hirsch's lecture where he was talking about mosquito control. Mosquito control was actually very good in South America when they had dictatorships. Unfortunately, kind of that slipped off with more democratization. So more dictatorships, less disease. Not sure that's necessarily such a good thing, but that may be some of that process, isn't it? People are used to these vaccines, and there's a lot less of this pro-disease movement in many of these places. Unfortunately, it's starting to spread, which is, again, rather an unfortunate case here. So this is a tweet which I saw just the other day, which to me is really kind of scary, particularly this last point right here. This is from, I didn't put the name here, a doctor who actually works in the refugee camps in Kenya. So, and I did, of course, retweet this appropriately, being what I do. So 2014, 2015, what happened? This is where you go to get measles, right? So there was a pretty major outbreak, end of 2014, beginning of 2015, where there were almost 1,000 cases. Fortunately, probably just statistically speaking, no direct deaths from this particular outbreak. Turned out to be traced to people who had been in Disneyland, and almost all of these cases were in people who were non-vaccinated. Sigh. But it was able to actually be traced back to an outbreak that was happening in the Philippines. You can look at the phylogenies, actually, of these different viruses, and clearly identify where some of these diseases have come from, but you can only identify them in these broader cases. What is interesting to me, if you think about whooping cough, on the other hand, that's pertussis. That's a bacteria, which has a much bigger genome. And I think, and we shall see, that people are trying to put together some of these genome sequences, and if somebody dies due to one of these pertussis strains that can be identified in someone who has willingly non-vaccinated their children, they may be liable. So genome sequencing may actually, I think, turn out to be very interesting, is one term for it, in terms of thinking about vaccines. So just another way of looking at this outbreak. Again, this was December 2014. Most of the cases were in California, but of course, lots of people like to go to Disneyland. And this ended up stretching out through a lot of the rest of the US. And just in case we thought this was just confined to 2014, 2015, this is those data here. This is actually the up-to-date numbers from the CDC. 2018, we actually already have 63 cases as of April. So it looks as if 2018 is actually probably also going to be a bad year. And these things, they vary quite a bit, but here 2004, 2007 was pretty low, and we're starting to see bump ups that are happening here. Some of those, unfortunately, are in regions of the country, particularly Minnesota, and that was this big 2008 bump here, where people decided not to vaccinate because of a fraudulent paper which was published in The Lancet and has since been retracted. And we can talk more about that too if people would like to, and if I can bring myself to actually talk about it. But again, so we have these relatively sporadic outbreaks. This is 2014. Again, this was the big Disneyland outbreak. But still, the numbers have gone up. And 2015, this year was 188, but 2016 was down here around 100, 2017 around 100, and 2018, I'm afraid, is probably going to be more up at some of these 2011 levels. That being said, again, almost all these cases are being imported from somewhere else in the world. Philippines sounds like a place where maybe there is going to be circulating measles, but also many of these outbreaks have come from places like France, Switzerland, England, etc. So not necessarily places where you would think that measles will be transmitted. One of the things that I forgot to mention about measles is it's one of the most transmissible virus diseases. They have forget what the actual term is for it, but basically one person can easily infect 18 other people. And it sticks around for at least a couple of hours. And so basically anyone who has been exposed within those couple of hours can be infected. And this can be in spaces as large as this room. So hey, we've got, how many people voted? 46. So if there were 2 people who had measles in here, all of us would have been exposed. Yeah?
So is it a candidate, like when we eventually vaccinate everyone against measles, would it be a candidate for the select agent list?
So the question is, would measles be a candidate for select agents? Wild-type measles is on the select agent list currently. You can only work with wild-type measles, I think at BSL-3, but many people actually work with it at BSL-4 because there is the vaccine, and so if you're vaccinated, you're fine. There are a few people who actually, once they have been vaccinated, particularly after the first time, are still susceptible to being infected, and that's why they brought in the second vaccine, which actually lowers that number. But still, you can have some people who are actually not then protected, which is another reason you want to have vaccine rates to be high. Other questions on this vaccine stuff? Yeah, Margo. What is the mechanism of requiring a second round? Oh, why do you need one as opposed to why it was mandated as such? Okay. Yeah, so why are you susceptible? That's a wonderful question. I don't know the exact answer to it, but if I remember correctly, I think in any given case, something like 95%, you will be protected. Then there's that 5% who won't be, and just statistically speaking. But if you then revaccinate everyone, it's going to be 5% of that 5%. So you've gone down even further. So that seems to be, that's my understanding of it. I'm not absolutely certain whether that's the case. And there are some people, those who are immune suppressed, et cetera, who can't be vaccinated. And so that's another reason, potentially also between the first vaccination and the second vaccination, which is when potentially some of these kids could be getting sick and being exposed. Yeah? I'm pretty sure that if you've had measles, then you are resistant to measles thereafter. It does seem to be, and as far as I can tell, it's lifelong immunity. There's some really interesting work going on, actually, at the West Campus of OHSU in terms of thinking about, how long immunity lasts. And it turns out for some vaccines, it's a lot longer than others. For measles, I don't know all the details on that. There's a fellow by the name of Mark Slifka who's done some really nice work looking at this. In fact, they've got about 70 years worth of data now on some vaccines. Of course, from the 50s, we're pushing that number now too. Yeah, there's a question here.
Yeah, so these outbreaks happen because that 95% ratio of people vaccinated is not just below that. So that 5% kind of safety for herd immunity, is that why these outbreaks are happening?
So the question again is a little bit getting a bit ahead of ourselves. I'll talk a little bit more about herd immunity when we talk about smallpox later on. But yeah, the question is basically, again, correct me if I'm wrong here, is the reason we're having outbreaks is because we're getting below this 95%. If I remember correctly, it's actually a lot lower than that 95%. And so many of these cases, again, in terms of the Disneyland outbreak, many of these people were just not vaccinated at all. And so that is much more of an issue, particularly true, again, of measles, because it's going to vary from disease to disease. If you've got something like measles that can spread to 1 to 18, then the threshold is going to be a lot lower in terms of being vaccinated to be able to protect everyone. But my understanding is that many of these cases you have vaccination rates of 60%, 50%, sometimes even lower than that. Take the case in the Somali immigrant population in Minnesota, where they had this outbreak in 8 and 11. The vaccination rates were below 50%. Okay, we ready to get 100% on another clicker question? Giving you all these options, right? Okay. And I'm told I should make harder ones, right? Not on Monday morning, not when everyone hasn't been fully caffeinated yet. Been good, I haven't been asking lots of obnoxious questions of people yet. Don't remind me.
People are exposed. Is there a difference in level of infection if they're younger versus older? If they're unvaccinated versus somebody that's 25 in good health versus a child who's 5, both get exposed. Do they both get the same type of infection or is one more severe?
Yeah, so the question that Nicole asked here up front is there a difference in terms of severity when you get infected, if you're younger versus when you're older? It does seem that the disease is worse when you're younger. That is complicated, of course, by the amount of vaccine that anyone has had, et cetera. So there's some issues with that. So it's not completely clear. And whoever this is messing with my statistics. I don't know what the idea is here. Say, what I have to do is just stop when we actually get to 100% here. So, okay, so that's all I have disease-wise. More questions on disease? Now we can talk about the interesting molecular stuff. Yes, finally, good. This does turn out to be important also in terms of thinking about how to treat some of these diseases as well. So, again, Paramyxoviruses are kind of these blobs, enveloped viruses, so not with a clear protein capsid on the outside. On the inside, however, they have these nucleocapsids. So we talked a little bit about nucleocapsids last time when we talked about enveloped viruses for the coronaviruses. This is just literally the genome plus protein. And if you break open these virions with a little bit of detergent, what you see are these structures, which hopefully are visible here, that are clearly helical around, and actually as we'll see, the RNA is actually wrapped around the protein in this particular case. Rhabdoviruses, you may wonder why we're talking about rhabdoviruses and paramyxoviruses in the same lecture, is that the structure of the virion is quite different, but the actual molecular basis of these viruses is extremely similar. And as we saw for rabies, their encephalitis is one of the major issues that happens. Unfortunately, as far as measles is concerned, it's a lot faster, so you can't vaccinate after you've been exposed. But this bullet shape actually turns out just to be this helical nucleocapsid, which is wrapped in yet another helix around this structure together with the infamous M protein. So let's see, we'll get back to our list now. Armand, what do M's usually stand for? Maturation protein is in MS2 and Q beta, but in the other ones we've talked about so far, what else does it stand for? It's not membrane, but that would be a good guess. Pardon? Confirmation. So these are often what they call the matrix proteins. We'll talk about matrix again. So what does matrix proteins do? What do they interact with, matrix proteins? Say in the coronaviruses, like we talked about last time. Capsid's one thing. What else? If you've got an enveloped, so nucleocapsid is genome. If you've got an envelope, what else is going to have want to interact with? What's in envelopes? Those will be your receptor binding proteins very often, or glycoproteins. So the matrix is usually sticking in between there. In the case of the rhabdoviruses, it's really just this M protein which gives you the shape. So it's the M protein itself. So if we look at the cartoon version of here, lo and behold, the M protein, the matrix protein between the membrane proteins and the nucleocapsid, again, which is this helical process. In the nucleocapsid, you have, again, incredibly creatively named N protein for nucleocapsid protein. And we'll see this for a lot of these other negative strand RNA viruses. But also because you have to have your own RNA-dependent RNA polymerase for all these negative strand viruses. Again, I haven't forgotten these are negative strands and talking about all those diseases. Negative strand polymerase also is associated here. And in the case of all of these paramyxoviruses, there's also this P protein, phosphoprotein, which helps the polymerase to do this polymerization process. So that's all nucleocapsid. You have the matrix in between, and then proteins on the outside. Hemagglutinin neuraminidase. This should sound very familiar because what does it do? It helps hemagglutinate and is your receptor binding protein. And then the F protein, again, ridiculously creatively named fusion protein. So these are what are all present on the outside of these paramyxoviruses, and also true for many of the rhabdoviruses as well. These fusion proteins, like we've seen in a number of other cases, they have a short hydrophobic peptide. That's what's shown here in gray. These hydrophobic peptides are almost always generated by a particular cellular process, which Travis might tell us about. Travis, Spencer, Jane? What cellular process might be generating these kinds of fusion peptides? Particularly important for maturation of virus particles. I'll give you a hint. Shall I give you a hint? The F protein is made from one gene, and it's got these two parts. So it is actually, in this case, it's not a polyprotein, but what happens to polyproteins? They get proteolyzed. What do you think is happening here? It's also being proteolyzed, exactly. So many, many, many of these fusion peptides are generated by proteolysis. Oops. So proteolysis here is what's leading to this fusion peptide. And one of the main reasons for this is you don't want to be having a fusion peptide sticking out on the outside of your virion as you're trying to make that virion and get it outside of the cell. So you're only going to want to have that fusion peptide exposed at the very end of the maturation process once it comes outside the cell. Yeah, Jacob. Okay, so the question is it a fusion process or a proteolytic process? What do you think? Okay, so I guess I didn't phrase it properly. So it's it's all part of that viral protein, and it's a cellular protease which cuts it. So there aren't viral proteases in this case, so it actually is kind of different than the case with the polyproteins that we saw in all of these positive strand RNA viruses. It's cellular proteases which are used to have this cleavage event. So maybe that was the confusion. Yeah? So this is all, the cleavage takes place all during maturation. So the virion on the outside is now primed again in your metastable state in such a way that the fusion peptide can bind. However, as you're making that particular fusion protein, you don't want this big hydrophobic piece because it'll just stuck to the cell that was producing that virus in the first place. Yeah? So I guess the question is just as it's being recorded is, doesn't this depend on you having lots of cellular proteases around to do this kind of maturation process? If you think about secreted proteins and signal peptidases, et cetera, there are lots of proteases, particularly in the endoplasmic reticulum and a lot of the secretory parts of the cell. And so the viruses seem to have evolved to take advantage of the fact that these are there. Yeah, so there doesn't seem to be a limit in terms of the proteases there. But when we talk about HIV, we'll talk about some other aspects of proteases. So stay tuned as far as that's concerned. So these are all happening, as I mentioned before, at the plasma membrane for all these Paramyxoviruses. Paramyxoviruses have this hemagglutinin neuraminidase and a separate fusion protein. So it's a separate gene that has this fusion protein. On the other hand, VSV has a combination of these two proteins. So the F protein and the hemagglutinin neuraminidase protein, known as the G protein, and it's not a GTP binding protein, it's a glycosylated protein. And we talked about these proteins on the outside of virions often being very heavily glycosylated. So that's the G protein. In the case of VSV, this binds to receptors on the outside of the cell, which we'll see turn out to be mostly sialic acid based, and then you have endocytosis. Instead of binding and fusing to the plasma membrane, this G protein is really, really good, I should say, VSV-G protein, in binding to all kinds of different cells. And so that's why it's so useful in terms of taking VSV and allowing it to infect lots of different cells. And that's why VSV has been used for so many cases of gene therapy, or also in the cases of some of these vaccine processes, because it can bind to and infect lots of different cells. So what comes in on the inside? It's the nucleocapsid. Again, once you fuse the membranes, pretty classic membrane fusion, hemifusion comes apart. Then the nucleocapsid is released. And the nucleocapsid here, remember, these are negative strand RNA genomes. So the negative strand RNA genome always has this nucleocapsid protein associated with it. And the RNA is actually on the outside, as I mentioned before when we looked at some of those EMs. And it turns out that each of these proteins, each one N protein, binds to 6 nucleotides. So 6 nucleotides, 6 nucleotides, 6 nucleotides, 6 nucleotides, 6 nucleotides. And that whole process allows you to package and protect that RNA from all of the hungry exonucleases which are floating around inside the cell. And the genome is always packaged. And the neat thing about this genome always being packaged, that can clearly differentiate it from any of the messenger RNAs. Messenger RNAs are not packaged. They don't associate with the nucleocapsid protein, so they can be translated. And so that's one of the main things that you see with these particular genomes. So, one more chance for everybody to get 100%? Yes? Good? We're all ready for this? This one may be a little harder, though. We should do 30 seconds and then let people chat.
Wait, are we allowed to chat now?
You can chat now if you like. It's all right.
Oh, that makes sense. Yeah.
Okay, so we're not at 90%. So tell your neighbors what you chose and why. Pardon? Yeah, no, that's why we're not. That's why I'm not counting them. Yeah, no. That's why we're going to do it again, and then it will count. Okay, ready to go? Much better this time? Okay, let's do it. You can see how much faster people vote, too. Sure.
Are you exchanging a protein that's originally on the rhabdovirus, or are you talking about using something of the CMV? Which one is active or?
So which of the rhabdovirus proteins was exchanged to make the vaccine? So, and I did not mention what that was, but most of you seem to think it was E as the G protein, but not for a number of reasons that I heard people talking about here, which is it binds to everything. Well, yes, it does, but if you exchange it, you're not going to be able to bind to lots of things and have it infect. However, if you have a glycoprotein, what's that interacting with? The host, and where is it in your virion? Outside. If you're trying to make a vaccine, where do you want things to interact with? On the outside. So yes, it is the E. I hear big sighs of relief here. So let's briefly talk about the fusion process. Again, this is pretty standard to what we've talked about before. Again, membrane fusion. In the case of the Paramyxoviruses, it's almost always at the plasma membrane. VSV is different, again, because it gets into endosomes. Sendai virus binds to sialic acid. Measles binds to various immune cell receptors. Again, these are the ones that love to bind to things on the outside of cells anyway. You need the hemagglutinin or the hemagglutinin neuraminidase, depends on your protein. F, again, structural changes, and many of these are trimers. So once you've done this fusion, this is the genome which comes inside the cell. And basically, these genomes, I haven't said Paramyxo or Rhabdovirus genomes here because they're practically identical to each other. Big difference is that the fusion and HN are the same. In these Rhabdoviruses, it's the G protein. So here again, now from 3' to 5', because we're negative strand viruses, starts with a short leader sequence. Then we have the part of the genome which encodes the N protein, nucleocapsid protein, PCV, matrix protein, fusion protein, etc. And then finally, the L protein, which is the polymerase. So here, major structural proteins are at the 3' end of the genome, but of course these are negative strand now. So one of the other critical things about these genomes is that between each of these protein coding segments is this intergenic region. And this is just shown in gray here. Each of these has this very well conserved sequence. The end of one of these messenger RNAs, this GAA sequence, and the start of the next one of these. Most important here is this stretch of U residues, which are present right here. U is going to base pair with what? A. Why might that be important? What do you need for messenger RNAs? You need caps and tails. What are tails? They're poly-A's. How do poly-A's happen? Turns out that these poly-A's happen through this process called stuttering. So, what is stuttering? It's the RNA-dependent RNA polymerase that when it gets to one of these intergenic regions, instead of just making one copy of that sets of U's, it makes many, many copies of those sets of U's. And then, in some cases, the RNA-dependent RNA polymerase falls off of the genome. And in some cases, it stays on and will start a new messenger RNA, get to the end of that, hit an intergenic region, make some poly-A tails, some cases continue, some cases fall off, etc. And in that process, you end up making a lot more messenger RNA for N than you do of PCV or of M or of F or HN, and then finally of L. So this solves the problem that we've talked about many times before. You need a lot more of the structural proteins than you do of the non-structural proteins. It's just statistically, certain percentage of the time, the dependent RNA polymerase will fall off here. Certain percent of the time, it will fall off here. Certain percent of the time, it will fall off here. One of the big questions that people had as far as how these negative strand RNA viruses made messenger RNA was whether the RNA-dependent RNA polymerase bound to the RNA at each of these intergenic regions, or it stayed on and didn't fall off. And the way that this was determined is that people made mutations in different parts of the genome and found if you made a mutation here, for instance, in the M protein, that was what we call a polar mutation, and it knocked out expression of all of these genes that were downstream of that. If, however, you'd had this multiple entry, just knocking out this gene would mean that you have just knocking out this gene. And this is discussed very nicely in TWIV 427. But again, the idea is here that the only place that your RNA-dependent RNA polymerase can bind is at the very end of the genome, coming in from solution, and then it's a dissociation process here. Between each of these, you can dissociate, you can dissociate, you can dissociate. Any question or just stretch?
Well,
Yeah.
no, a similar thing is happening in the PCV genome region with the extra genes being added to the genome.
Yes, it is, and we're probably actually going to talk about that on Wednesday because it turns out to be an important thing to talk about, and even though we're not going to get to it today because it's about 10:04, but we'll definitely talk about it. It does tend to be a very similar process which is happening there. But before we all run away, I did want to talk about one more thing, which is the difference between replication and transcription. So making genome versus making messenger RNA. And this has to do with the concentration of the N protein. So at low N protein concentrations, you make messenger RNA. At high N concentrations, you make genome. And we'll start here on Wednesday in lecture and continue with this. So unlike in many cases, the slides at the end we won't get to, these ones we actually will get to because they're really important also for thinking about filovirus replication in Wednesday's lecture.